next presentation is from Henri Dumont, who is working with Dr. Jonathan Trent, and will be telling us about anti-tumor effects of Notch signaling pathway in gastrointestinal stromal tumors.
It's my pleasure to be here today to present my data about Notch pathway in GIST. GIST is a gastrointestinal stromal tumor. It's in fact the most common mesenchymal tumor of the GI tract. This disease is driven by a gain of function mutation of KIT or PDGFR alpha. This disease is fascinating because it acts like a paradigm. We came from 10 years ago where this disease was poorly understood, where the biology was not known, that was very resistant to chemotherapy, to now the utilization of tyrosine kinase inhibitor that changed dramatically the history of this disease. But most of the patients will develop resistances, mainly caused by a second mutation of KIT or PDGFR alpha. The scope of this work is to find alternative strategy to treat patients after imatinib failure. What is the cell of origin of GIST? It's the interstitial cell of Cashel. The interstitial cell of Cashel is the pacemaker of the GI tract. They share common feature with neuroectodermal cells. In this kind of cells, neuroectodermal cells, there is one pathway that is key. It's the Notch pathway. So we make the hypothesis that addressing the role of Notch in GIST could be interesting to understand more the biology and to find potential therapeutic strategies. I will give you an oversimplified view of the Notch pathway. The activation of this pathway starts by the interaction between the ligand and the receptor, following by a cleavage by metalloprotease Adams and by the gamma secretase complex that will release the intracellular domain of Notch into the cytoplasm. This intracellular domain of Notch will translocate to the nucleus and will convert the CSL complex from a repressor to an activator, and will activate the target genes, mainly the S family members. To investigate the role of Notch pathway in GIST, we first transduce our cells with the active form of Notch, the intracellular domain of Notch, and we followed the GFP expression after days after the transduction. And we found that at the end, for example here at day 12, the population that express ICA, intracellular domain of Notch, disappear. We did in parallel the same thing with one of the main target genes of Notch, that is S1, and we observed also a decrease in viability, but not so profound that with ICN1, meaning that there is not a fully recapitulation of the Notch1 effect. To see if this finding is relevant clinically, we study 15 GIST patient sample, pre-treatment, and by a Kaplan-Meier analysis, we found that the expression of S1 strongly correlates with the relapse. Regarding the previous finding, we have a therapeutic opportunity because one class of drugs are known to upregulate Notch. It's the HDAC inhibitor, and in particular SAHA. If we can upregulate Notch, we make the hypothesis that we will decrease the viability of the cell. And indeed, SAHA, this HDAC inhibitor, upregulates Notch1 at the mRNA level and at the protein level. Additionally, SAHA and decrease the cell viability, increase the apoptosis, showing by increase of the subsequent population by cell cycle analysis, decrease the activity of KIT, shown by Western blot, decrease of the phospho-KIT, and the downstream pathway, phospho-ERK in this case, and decrease the total KIT expression by flow cytometry. If the mechanism of action of SAR is really due in part by this upregulation of Notch, knockdown of Notch should rescue the cell from the treatment of SAR. We use a dominant negative construct for S1, and we found partial rescue in terms of cell growth after SAR treatment. We found also rescue the KIT expression. Here it's vector alone compared to the dominant negative S1, where we have a partial rescue. To give you a summary about Notch GIST, we have found that Notch1 acts as a tumor suppressor in GIST in vitro, that S1 seems to predict the clinical outcome in GIST. We need to work on a bigger sample, and we are planning to use also tissue microarray to see at the protein level if we can find the same result. And we have finally found that activation of Notch1 is possible by SAHA. The next step will be to confirm this result in vivo model, because the in vitro model it's not enough. To date, there was no good in vivo model for GIST, only subcutaneous injection of GIST cell. We decided to create an orthotopics models by injection of cells into the gastric wall of the skip mouse. The cells are forced to express 5-fold luciferase that allow us to see the tumor by non-invasive imaging. I would like to thank my two mentors, Dr. Trent and Dr. Hughes, and all the members of both labs, in particular David Renanzo, Yasmin Yang, and Norman Yang, Yasmin Jabber, and Kenji Oko for the in vivo study. Thank you very much. I will not forget to mention Sarah Jono, my wife. Thank you.